Good morning, Panchika. Okay, so I'm Rahul Saha and we'll be proceeding with the same worksheet that we had commenced last class. Good morning, Tarun. So, come directly to this question number. So, we had finished till, I think, question number 27. Okay, so question number 28 is pretty easy. A human female with Turner syndrome, she has X naught, right? So she will have 45 chromosomes with X naught. So there is no extra X chromosome in the case of Turner syndrome. So it is again a specific feature of non-disjunction, right? Nothing but non-disjunction, which you already know completely. So in the meiosis, suppose in that female, one of the cells would be having both the X chromosomes, okay? One would be having no X chromosome here, 0X. And then specifically that ovum might have mated with the sperm, and sperm would be containing an X chromosome. So it is the result is X, but here nothing, so X naught you can say. So coming back to the question number 29 now. Select the incorrect statement with regard to hemophilia. So it is again an easy question, right? So try it out. Select the incorrect statement with regards to hemophilia. So it is not a dominant disease. Does it look like a dominant disease? It's a sex-linked recessive disease, right? So not at all. First option only if you read it's a dominant disease. It's not a dominant disease. A single protein is involved. It is correct. You can have protein factor 8. You can have protein factor 9 deficiency. You can have protein factor 5 deficiency. Okay, hemophilia A, B, C. Then parahemophilia protein factor 11. So that is correct. Sex linked, it is correct. Recessive, it is correct. So since it's a dominant disease, is the first option. It is not at all a dominant disease. It's a sex linked recessive disease. So it is completely incorrect. Okay, first option. Next, if we talk about which of the following cannot be detected in a developing fetus by amniocentesis. So I think you are aware of amniocentesis. So this you can say theory comes in, you can say, your chapter number 4, I think, yeah. So that is known as amniocentesis. So you are all aware of the amniotic fluid, right? In case of amniocentesis, what is done is your amniotic fluid is being checked, right? It is being checked for, or it's a procedure you can say during the pregnancy, where a part or a minuscule volume of amniotic fluid is taken out. First of all, okay. It is also referred to as simply amniotic fluid test. Okay, it is used in prenatal diagnosis of chromosomal abnormalities, fetal infection, as well as sex determination. Okay, amniocentesis. If we draw amniotic fluid, then you can go for karyotyping, chromosomal disorder. Then you can go for infections, any kind of viral, bacterial, fungal infections. Okay, and also since you are going for karyotyping, you will also get to know the sex of the fetus, so sex determination. Okay, these things can be easily done via amniocentesis, but this is banned, right? Sex determination is banned. If you can go for, you can say abortion if you have really tumultuous chromosomal disorder, because it's not correct. It's not, you can say, wise to give birth to a child, a progeny with severe chromosomal deficiency. Okay, because the treatment is not there as per the present technology. Even if you go for CRISPR-Cas9 gene therapy, you won't be able to treat him or her, and she'll suffer for the rest of his or her life. You will suffer also along with the child. So if this is there, you can abort, but this is banned. Now the question is, which one of the following cannot be detected? So Down syndrome is a chromosomal defect, can be easily detected by amniocentesis. 21st chromosome trisomy. Klinefelter double XY can be detected. Sex of the fetus can be detected. But jaundice is a metabolic disease. Okay, it's a metabolic disease. Bilirubin accumulation maybe liver infection. So those cannot be detected. Metabolic diseases cannot be detected, like hypertension, diabetes, hyperlipidemia, hypercholesterolemia. You cannot detect all these diseases, glomerulonephritis. So you cannot go for the detection of metabolic diseases via amniocentesis. Question number 30. Now 31. If both parents are carriers for thalassemia, so I have told you in the previous classes also, we have two types of thalassemia. Improper hemoglobin synthesis, we have alpha thalassemia, beta thalassemia. And thalassemia is nothing but autosomal recessive disease. Autosomal recessive disease because the genes are present on the autosomes, not on the allosomes or sex chromosomes. Fine. Now, if both the parents are they're wanting to know, they haven't put here first of all alpha or beta. So alpha, if it happens, so small a must be the case. Okay, if it is beta thalassemia, then small b shall be the case. If in case of alpha, one is dominant, means one is the proper gene. Okay, you will be phenotypically normal in this case. You will be phenotypically normal in case of beta also. If one is normal, one is recessive, means you can say possessing nonsense or missense mutations, right? So this and this genotype will give you normal phenotype. So only you will be in the diseased phenotype in these two genotypes. So disease phenotype means either alpha thalassemia or beta thalassemia. Here beta, here alpha. Okay, now come to the question. Since you know about this, now come to the question. If both parents are carriers for thalassemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in an affected child? You can assume any of these. Okay, both parents are carriers means, suppose I am checking for alpha thalassemia since it is not given here specifically. So AA, go for crossing. Pretty simple. Okay, don't get perplexed in these questions. It's pretty simple. Okay, so what will happen? The child getting affected is only 25%. Okay, but the carriers are 50% here. Again, you will develop 50% carrier. Only again 25% will be no carrier and again not affected whatsoever. So this will be how you are going to solve this question, right? Now question number 32. If two persons with AB blood group marry, okay, and have sufficiently large number of children, these children could be classified as A blood group B 1:1 ratio. Modern technique of protein electrophoresis reveals presence of both A and B type proteins in AB blood group individuals. Okay, the thing is, in these types of questions, listen very carefully. These the first two lines are just making you perplexed. There is no use of this 1:1 ratio. Okay, the reading of the first two lines has got nothing to do with the solution of the question, or it doesn't impact in any manner whatsoever how the solution is gonna be decided. Okay, so only these last two lines are important. These last two lines are only important. Modern technique of electrophoresis reveals, okay, presence of both A and B type proteins in AB blood group individuals, which we already are aware of. Right, I have shown you multiple times its substance. Then blood group A, in multiple cases I have showed you that blood group A is what? Blood group A means to the last galactose, okay, which has also fucose attached. You will have N-acetyl galactosamine. Okay, blood group B to the last galactose of the S substance, again fucose attached. You will be having another galactose. Okay, so this enzyme which goes for the covalent attachment, that is N-acetyl galactosamine transferase, this galactosyl transferase is present in the Golgi apparatus, is coded by the IA gene, is coded by the IB gene. Now, if both these genes are present in an individual like this, in a homologous copy, you have IA, its homolog, okay, contains IB, then both will be expressed. So what is it? It is nothing but simply codominance. Okay, Just read the last two lines of this very question. Don't be perplexed about one-to-one ratio. Now, what I have to do? I have to start with a cross. No. This is an example of direct codominance. Okay, that's all you need to know. Question number 23. Okay, sorry, 33. Question number 33. So when we come to question number, good morning, Asma, good morning, Ayushi. So when we come to question number 33, okay, in question number 33, which idea is depicted by a cross in which F1 generation resembles both the parents? When you are resembling both the parents, what you are doing? Right? Resembling both the parents. Question number 33. It is again a very very easy question, but little bit concept is needed. Again, inheritance of one gene, no. Incomplete dominance, no, because you are resembling nothing then, right? Incomplete dominance, none of the parents. Complete dominance, you will be resembling only one. Resembling both means, suppose one has blood group A, one has blood group B, so I am resembling blood AB, I am showing resembling both the parents. That means it is again an exemplar, a paradigm of codominance only. Resembling both the parents, that is nothing but codominance. Okay, coming to question number 34, very very good question. Which one is the correct statement, okay, with regards to the importance of pedigree analysis? You've already learned pedigree analysis. I had given you the tips also how to solve. Okay, now let's check the options. It confirms that DNA is the carrier of genetic information. It helps to understand whether the trait in question is dominant or recessive. That seems completely correct to me. Then it also lets you can say, assess, or it confirms that the trait is linked to one of the autosome, correct. It helps you trace the inheritance of a specific trait, that is also correct. So what happens is, pedigree analysis is used for what? To analyze the pattern of inheritance of a particular trait throughout a family, right? The pedigree shows the presence or absence of a trait as it relates to the, you can say, relationship among parents or among offsprings or among the siblings. So by analyzing pedigree, we do what? We can determine genotypes, even identify the phenotypes, predict how a trait will be passed on in the future generation also. So you can know whether the how the alleles have been inherited, okay, whether they are dominant, recessive, autosomal, mitochondrial inheritance also, which is not in your syllabus, then sex linked, okay. But you cannot know by any means whether it is confirming that DNA is carrying the genes, okay. So it is not sequencing the DNA that what all genes are present in the DNA, okay. It is not sequencing the DNA that it will tell you that this is a chromosome, okay. This is the P arm, petite, Q arm, Q long arm. It will tell you that in this locus of the chromosome this specific gene is present, okay. Suppose gene for insulin is present, okay. It cannot specifically tell you. This is not a genetic sequence technique, okay. It is not gene sequencing, it is not DNA sequencing technique, okay. That is the truth here, okay. It is not Maxwell Gilbert method or something like that. It is simply a pedigree analysis that we just look at the inheritance pattern and can predict the probability of that disease happening in the specific, you can say, offspring slash progeny or not, okay. So option A is the correct one. Question number 35. Down syndrome is due to, many times we know, three copies of chromosome number 21. Trisomy in 21st chromosome. Again, non-disjunction is the answer here. So we have discussed many a times about non-disjunction and how it can give rise to your Down's syndrome. Okay. So that is your Down syndrome here. Okay. Now coming on to question number 36, which is a very, very good question. These kinds of questions can perplex you in the PMT examination or NEET examination. Now, how to solve this question? A normal visioned man, okay, whose father was colorblind. Okay, since he is a normal visioned man, okay, and color blindness is again a sex-linked recessive disease that you should know. Okay, which kind of disease? What? Otherwise, if you go with the autosomal trait here, you will be in a lot of problem and you cannot solve this specific question, right? So color blindness, normal vision man. Father was color blind, doesn't matter. If he is normal vision man, X-linked recessive, then he will be XY, no problem whatsoever. He is not carrying, you can say, the genes for the color blindness whatsoever. Then he marries a woman whose father was also color blind. Okay, so the thing is, I told you, from only father to daughter. Okay, it can be easily transmitted. Father to son is pretty, you can say, difficult, but it also gets transmitted. The thing is, if he's marrying a woman, okay, whose father was also color blind, so we can generally take this as a starting point that the woman is XC, carrier for color blindness. Okay, this is the, sorry, woman. The woman is, you can say, carrier for color blindness. She is the carrier for color blindness. Now, if you go for the, you can say, trait, or if you even start before that, like the parents, they are saying that you have a normal man and a carrier woman. Okay, you have a normal man and a carrier woman, and then when you talk about the gametes will be one, the gametes X, one of the gametes Y, one of the gametes XC, one of the gametes will be X, right? And then when we cross this, okay, then when we cross this specificities, okay, XY, then XCX, let's cross it. You will get XXC, XCY, okay. Then you will get XX completely normal, then you will get XY. So what are you getting here? Okay, this one, since it was carrier here, but XCY will give you color blind son, right? Color blind sun. Okay, XY is completely normal. Again, XXC is completely normal phenotypically. This one is completely normal phenotypically. Okay, so after looking at the crosses, what can I say? The daughter born to them can be either normal or carrier to the disease. Okay, but the possibility, okay, the question says what are the chances of this child, okay, if the first child is a daughter, the possibility would be completely a 0% possibility. There wouldn't be any possibility whatsoever. Okay, in question number 36. Now coming to question number, you can say, 37. F2 generation in a Mendelian cross showed that both genotypic and phenotypic ratios are 1 to 1. Okay, 37th question. So it represents a case of not codominance. Okay, it doesn't represent the case of, you can say, dihybrid cross also. Okay, listen to this question very carefully. So F2 generation in a Mendelian cross. Okay, in a Mendelian cross, it showed that, okay, it showed that both genotypic and phenotypic ratios are same, very important, are same. Here it is showing that they are completely same, that is 1 to 1. Now it represents the case of, now what you can see here is, okay, the correct answer will be monohybrid cross with incomplete dominance. But how? So incomplete dominance is a type of intermediate inheritance, we know, in which one allele is not completely dominant over its paired allele. We have already seen the Snapdragon thing. I had also explained to you, okay, the mechanism behind it. Because the thing is, when you come to gene quantification, that is the mechanism. Gene quantification. That we know that if these two are the genes, okay, homologous chromosomes. Suppose this one is the proper one, this one is the improper one, defective one, mutational one, missense or nonsense mutation possessing one. What will happen is this is a double stranded DNA, this is a double stranded DNA. So one time this can be opened up by RNA polymerase, okay, two, and you can get mRNA from here. In another event, you can get mRNA from here. Doesn't matter that it's not a chance of probability. You can say probability that all the time this specific DNA will only be used for mRNA production. This can also be used. That when this is used, you get zero protein or a dysfunctional protein or a dysfunctional protein. So the quantity of protein is not enough, okay, because it is only produced by the heterozygote condition, only one allele, that it will be able to completely shift the effect, phenotypic effect towards the normal parent. Okay, that is known as incomplete dominance here. Okay, so the thing is, in this question, what happens? The third phenotype, okay, is observed in only the heterozygous condition, right? So in monohybrid cross with incomplete dominance, okay, the phenotypic and genotypic ratio is always 1 to 1. Always remember, it is always 1 to 1. You can check this out. Okay, phenotypic and genotypic ratio are both 1 to 1 here. Okay, you can see. Suppose I am crossing this one with this one. Okay, suppose I am doing it. Okay, so what I'll be getting is monohybrid cross. Okay, so A, so A, then here capital A, capital A, small a, small a, small a. But what you would be seeing is, suppose this is white, this is black, like this. Suppose this is red, this is white. So you'll be thinking all three must be red. No, genotypic ratio here is 1 to 1. Phenotypic ratio will be same. This is suppose red, so these two will be pink. This is white. So phenotypic ratio is also the same as genotypic ratio. So the answer is monohybrid cross. Okay, not dihybrid. It's a monohybrid cross with incomplete dominance. Always remember this is the same phenotypic ratio. Okay, so it's a tricky question. Phenotypic ratio in a monohybrid cross is equal to genotypic ratio in case of incomplete dominance. Okay, genotypic ratio in case of incomplete dominance. Very, very important. Okay, now next question. Now come to the next question. Question number 38. Okay, represented below is the inheritance pattern of a certain type of trait in humans. Okay, which one of the following conditions could be an example of this pattern? Okay, so female father to daughter, okay, male father to daughter, then male or female mother to son. So it's a very, very easy thing. Okay, it means it is a sex linked trait. Very, very easy. It's a sex linked trait. And sex linked trait is which one here? Sickle cell anemia, no, autosomal recessive. Okay. Then phenylketonuria, autosomal recessive. Thalassemia, autosomal recessive, meaning that you can say only the sex linked is hemophilia, which is sex linked recessive. It must be hemophilia. Very, very easy. When it is showing from mother, father, son, daughter, means it is related to sex linked. Okay, so it is quite easy here. Okay, don't get confused or perplexed whatsoever. Now the thing is, a test cross is carried out too. So multiple times we have talked about the test crosses. A test cross is carried out to do what? So what is a test cross? When we have something known as, you want to check, okay, whether this is this one or this is this one. So what you do is you just cross it with a homozygous recessive trait. So here the phenotype will be the same. Here you can say the phenotype will be 50-50%. Okay, so the phenotype will change. Okay, so this is what test cross is all about. So what it does, so Mendel developed a system of conducting a verification of the results obtained by him. It is known as the test cross. It is a cross between, you can say, F1 plant and the recessive parent. So what it does is a test cross conducted for the monohybrid inheritance results in the two opposite characteristics, right? Expressing in the ratio 1 is to 1. Okay. Now you can do multiple things, but the thing is, what does it, the real thing, what it is intended for is to see what is the genotype of a plant. Okay, genotype of a plant at the F2 generation. So what is the genotype of the plant at the F2 generation? So this is what a test cross really aims at. Okay, whether it is homozygous dominant or a heterozygous condition, depending on this result 1 is to 1 or completely, you can say, dominant phenotype. So that is the meaning of, you can say, the test cross. Now coming on to the question number 40. Now, when two unrelated individuals or lines are crossed, okay, listen to this question very, very carefully. When two unrelated, unrelated means they are not related, okay, individuals or lines are crossed, the performance of F1 hybrid, okay, the performance of F1 hybrid is often superior to both its parents. Okay, it's often superior to both its parents. This phenomenon is called as. So the thing is, it is not called as transformation, splicing. We know only metamorphosis. It is in developmental biology course that I'll explain it to you in little bit in the human reproduction chapter only. This is being done right now. The thing is, the answer is heterosis. Now, what is heterosis? Now, heterosis is a condition, okay, which is not given in NCERT, but it comes in NEET examination. So, heterosis, so just listen to it. Heterosis is a condition which refers to, which refers to, you can say, the phenomenon that progeny of diverse varieties of a species or crosses between species. Here what is being done, crosses between species. It will do what? It will show increase in growth, size, fecundity, function, increase in growth, size, function, okay, yield or other characters, say yield or other characters in hybrids, okay, over those of the parents. So that is the, in hybrids over those of the parents. So if this is true, okay, this is known as heterosis. Okay, so it's a very, very special phenomenon that the hybrid, okay, heterosis means the hybrid is able to, you can say, become better than the homozygote or homozygous conditions. Okay, so that is known as heterosis. Now, you may be, you can say, interested to know that how does it happen. Okay, so what they do is they keep on exploiting the, specifically the plant breeders, okay, and also animal breeders also. Sometimes they exploit heterosis by mating two different pure bred lines that have certain desirable traits, okay, that have certain desirable traits, very very important, because when these two combine, those traits get combined via, you can say, genetic crossing, and it becomes much better. Okay, it becomes as a, you can say, much better way to get the, you can say, phenotype in the real world, which is better than, you can say, both the parents. Okay, when it comes to the economic importance, when it comes to the genetic engineering importance, so that is always the case in case of heterosis. Okay, so the thing is, it is also known as hybrid vigor outbreeding. Okay, so again, the phenomenon which causes crossbred individuals, you can say, to have an increased level of, you can say, performance for certain traits over the above, you can say, average, you can say, performance of their straight bred parents. Okay, so that is known as heterosis. So the thing is, there are two postulates also. If you see postulates molecular biology wise, so you have dominance hypothesis of heterosis. Okay, because if I just say, it may not be ringing the bell. So dominance hypothesis is present in heterosis, and you have over dominance hypothesis of heterosis also. There are two postulates: over dominance hypothesis. So dominance hypothesis holds that increased vigor and size in a hybrid is due to the combination of favorable growth genes by crossing, you can say, two inbred ones. Okay, so it is the interaction of dominant factors. You can say a polygenic effect of the dominant genes. You can say it is just like that dominant hypothesis. Whereas over dominance hypothesis, there is a physiological stimulus to the development that increases with the diversity of the uniting gametes, you can say. So it is known as single gene heterosis also. Okay, because two alleles it got included here, and because of the inclusion, you can say it became over dominant. Okay, or you can say super dominant. It is like that, super dominant. You are becoming super dominant after the inclusion of both different alleles from different lines together completely. So if you have this example, okay, so I'll just show it to you one example here. Suppose a parent has AA, okay, BB, CC, DD genotype. Wait, I'll change it up. Suppose we are taking four genes, okay, CCDD. It is crossed with opposite one, AA, BB, capital C, capital C, capital D, capital D. Okay. So suppose I am giving the phenotypic ratio to it. I am assigning one and half, one and half. Okay. Plus one plus one. This is total five. This is also one and half, one and half, total 5. Okay, but when the two will combine, what you will get is AA, BB, CC, DD. So here super dominance is happening. Each will be assigned now double phenotypic ratio. Okay, so you can say 2 plus 2 plus 2 plus 2. Now they are becoming 8, much better than, you can say, 5. Okay, so that is known as super dominance, or you can say over dominance phenomenon. Okay, in the F1 crossing, excessive expression is happening. Expression has increased. Okay, it is the super dominance hypothesis related to heterosis. Now coming on to this next question, which of the following conditions, question of 41, correctly describes the manner of sex determination. Okay, question number 41. If you check it out, homozygous sex chromosome ZZ. Okay, define female sex in birds. That is female heterogamity, so it must be ZW. So not ZZ. It must be ZW. Then you can say X naught of sex chromosome determines male sex in grasshopper. X naught condition in humans is found in Turner syndrome, determines female sex. Okay, so homozygous sex chromosome XX produce male in Drosophila. Okay, so the thing is you need to understand the sex determination. Okay, the kind of sex determination which happens in Drosophila also, and also you can say the sex determination happening in birds, and then you already know that the sex determination done by the Turner syndrome. So X naught is not really a sex, right? It is a completely contruded one. Okay, so it doesn't hold up anywhere, right? So now coming on to the sex determination in birds, first of all. Now sex determination in birds, if you go for, what will happen is you can check properly sex determination in birds. Okay, so the thing is you have the ZZ type. Okay, you have the ZZ type and the ZW type. You have both in case of birds, sex determination in birds. So ZZ and ZW. So what happens is when you talk about the ZZ type, what are you getting in the ZZ type? So you have sex determination in birds. So you can see here the sex determination in birds. If I write here, let's start with the first option. In birds. So the ZZ is taken as male, the ZW, okay, is taken as the female. So not at all. This cannot occur. Okay. The first option went out of the window completely. Okay. The first option went out of the window. Now you have something known as the second option. The second option is, or let's come to the third option first. X naught as the Turner syndrome. Okay. Or X naught as the, okay, let's come to second option. Yeah, X naught sex determination in case of male sex in grasshopper. Okay, so the thing is, you can say a sex determination pattern must be followed properly. So the thing is, in X naught type of sex determination, the female produces two gametes with X chromosomes, while the male produces only one X chromosome. That is the X naught type. If you consider, okay, that I had taught you also. The female will produce both. Male produces only one, okay, X chromosomes. And the thing is, the gamete with X, okay, fuses to form Drosophila, okay, fuses to form Drosophila, like this one. So if I say the female here, female sign XX, this one is X naught. So the once the gamete happens, the gamete will be X and X. Here the gamete will be X, nothing. So when you cross it up, okay, when you cross it up, X X, you have X here, nothing, right? So X X, when you have X X, you have also, since here it is nothing, so you will have X naught here. So this X naught, okay, is the, you can say, the male sex in grasshopper. Is the male sex in case of grasshopper, because the X naught type of sex division is used there. Okay, so that is the correct option. And once you come to the sex determination in Drosophila, okay, sex determination in Drosophila is little bit, you can say, confusing. So I'll tell you sex determination in Drosophila also. In Drosophila melanogaster is little bit confusing. What you have is, okay, it is not the, you can say, perpetuation of X chromosome to autosome. Okay, it is the ratio of X chromosome to autosome. Always remember, the ratio matters in Drosophila. Okay, the ratio always. So X to A ratio it is known as. X to A ratio matters in case of Drosophila. Okay, that's why it becomes more challenging. Suppose you have three X chromosome, okay, and two autosomes, okay, like that. So what will happen is number of, you can say, X chromosomes and number of sets of autosomes. So suppose number of X chromosomes to the number of sets of autosomes. That is clear in Drosophila. So what happens is if you have the number of sex chromosomes equal to 3, number of autosome sets in Drosophila is 2, okay, you are getting X to A ratio, that is 1.5, okay, here you are getting 1.5. So that will make it a meta female, okay. Then 4 to 3 and all that, so you can get a male, okay, you can get a normal female, then meta male, like that. So you will get a normal female when this is the ratio 2 is to 2, that will give you a normal female, like that. So this is how the sex is determined in case of Drosophila. So it is kind of, you can say, little bit confusing in that sense, that sex determination in Drosophila, okay, that is, that depends upon, you can say, the ratio of the chromosomes or the ratio of the autosome to chromosome, okay, rather than the dependency. Okay, so what you have is, if you check it another way, so chromosomes, okay, like this. So if you have 4x chromosomes in Drosophila, and that is 4 autosomes, so ratio is 1 ratio 1, 1 ratio 1. So again you will get a normal female here, normal female. Okay, again if the ratio is 1, if you have 3x chromosomes, just check it out. That is, again ratio is again 1, again female. Okay, so that is how you get females in Drosophila if the ratio is 1. Okay, if the ratio is like this, suppose it has XY, is only one set, okay, X to A will become 0.5, then when the ratio is 0.5 only, then you get a normal male. But you must have Y chromosome, okay, because in this case also you can get 0.5 if you have X and A, since there is no Y, okay, 0, so again 0.5, but you will get a sterile male in this case, you will get a sterile male in this case, in case of Drosophila. So that differs from condition to condition. Now, which of the following conditions of the zygotic cell would lead to the birth of a normal female child? Okay, question number 42. So, very easy. That must be, you can say, zygotic cell. Which of the conditions of the zygotic cell would lead to the birth of a normal female, so a normal human female child, means 2x chromosomes. Nothing to think about here, right? 2x chromosomes. Now, test cross in Drosophila. Question number 43. So must be done any organism you take. Doesn't matter. Don't get confused that Drosophila human, what will happen. Test cross is a test cross. The F1 hybrid with a double recessive genotype. That is what is test cross. F1 hybrid with a double recessive genotype. Nothing else. Okay. So now again question number 44. Which one of the following symbols and its representation used in human pedigree analysis is correct? Okay. So again, it's a very easy thing. Which one is the correct one? So Unaffected male female is just the opposite, it is incorrect. Okay, so the consanguineous one, related ones is the correct one. So A is the correct one here in question number 44. Question number 45, if you check, ABO blood group in humans is controlled by the gene I. Three alleles IA, IB, and IO, or you can say small I. Since there are three different alleles, six different phenotypes are possible. Okay, but how many phenotypes can occur? How many occurs? Four only, right? You have AB blood group, A blood group, B blood group, and O blood group. Only four occur because of codominance between AB. Okay. So coming to the 46th one, 46, you can say question. Select the correct statement from the ones given below with respect to dihybrid cross. Okay. Tightly linked genes are the same chromosome, so higher recombination. Genes far apart on the same chromosome, so very few recombination. Genes loosely linked on the same chromosome, so similar recombination. Okay. Then tightly linked genes are the same chromosome, so very few recombinations. Okay. So if you are linked highly, I have told you very easy thing. Linkage is inversely proportional to recombination. This was the general law that I had given to you. Okay, so means if the genes are too close, okay, genes A and B, SPO11 enzyme, RUB enzyme, RECT-BCD in the prokaryotes, RAT51 in the eukaryotes, they won't be able to cleave very easily. You need to be far apart for recombination to happen more, you can say, profoundly. So option D is tightly linked genes on the same chromosome, so very few recombinations. That is the correct option in case of 46. Now coming to 47th question. Okay. The genotype of plants showing the dominant phenotype can be determined by very easy test cross. Okay. Again, that is the test cross. Question number 48. Which one of the following cannot be explained on the basis of Mendel's law of dominance? Mendel's law of dominance. The discrete unit controlling a particular character is called a factor. It is correct. Out of one pair of factors, one is dominant, other is recessive. That is correct. Alleles do not show any blending, and both characters recover as such in F2 generation. This cannot be explained because codominance, incomplete dominance, those things cannot be explained by Gregor-Johann Mendel's crossing. Okay, when you get like you got 1:2:1 ratio. In case of incomplete dominance, okay, in the F2 generation of the monohybrid cross, did it justify? Because it didn't coincide with a 3:1 phenotypic ratio, right? You got a phenotypic ratio as 1:1. So it didn't coincide with phenotypic ratio of 3:1. So it is a conflict, it is a contradictory thing as far as Mendel is concerned. Okay. Then coming on to question number 49, ABO blood grouping is controlled by I gene. Okay, that is done. That is again a repetitive question, I think 4. Question number 50. So the pedigree chart is not given here. Okay, so come to question number 51 then. Okay, so a cross in which an organism showing a dominant phenotype, okay, a dominant phenotype is crossed with the recessive parent in order to know its genotype. Okay, 51 is known as again a test cross. Various ways you can see how many questions are asked in test cross in a twisted pattern. This is again test cross. Okay. Now in antirrhenium, two plants with pink flowers are hybridized. Okay. Question number 52. The F1 plants produced red, pink, and white flowers in the proportion of 1:1. Again, incomplete dominance. Okay. What is the genotype of the two plants used for hybridization? Okay. So genotype must be this only. 1:1 you are getting. Genotype must be this RR. Then only 1:1 you will get, right. This thing you are getting. This is showing the same phenotype. So 1:1 is the phenotypic ratio, same as the genotypic ratio, whereas PR and GR genotypic ratio are completely the same. So capital R, small r is the correct answer for question number 52. Now Select the incorrect statement from the following. Question number 53. Okay. So incomplete, I want to know. So number one is, you can say, galactosemia. Okay, galactosemia is an inborn error of metabolism. Okay, so galactosemia is what? Galactose in the blood. Okay, incorrect statement. So it is an inherent error. Okay, it refers to a group of inherited disorders which impair the body's ability to process and produce energy from a sugar known as galactose. It is known as galactosemia. So undigested sugars, they build up in the blood. That is correct. Now, small population size results in random genetic drift. That is completely correct. Okay. Now, when you talk about baldness, okay. So the thing is, question number 53. If you check baldness, okay, just check the question option number C. Baldness is a sex limited trait. Okay. Do you think it is correct? Okay. So the thing is, linkage is an exception, question number D, to the principle of independent assortment in heredity. That is correct. So baldness sometimes, okay, can occur due to, you can say, in case of females also. Sometimes it can occur. Okay. So if a male has one recessive allele, he'll show that trait. Okay. But you can say if the female has two recessive traits for the same, they will show the same trait, but that is a very very rare condition. Okay, don't think it is like that. So you just, if you check the allele, so a male, if it has one allele only, if it has one allele only, okay, so it will be showing it. Okay, one allele will be sufficient for males to show one allele, but if the females have got both the alleles, then they will show. That is the condition in case of baldness. It is not a sex limited trait. Okay, it is an incorrect option. Okay, only thing is, one allele is enough for the males to show, but that same allele must be present in the homozygous condition, okay, for the females to show baldness. Okay, now question number 54. Sickle cell anemia is caused by what? Caused by a substitution of, you can say, valine by glutamic acid? No. Okay, it is not substitution of valine by glutamic acid. Characterized by elongated sickle cell within a nucleus. Okay, autosomal recessive dominant trait? No. Okay, what happens is, there is no nucleus, right, in the RBCs, so this option is correct. Dominant is correct. This is correct. This is also, A is incorrect, whereas glutamic acid is substituted by valine in the sixth position of beta chain. So the option B you can consider to be correct, because it's caused by a single base pair mutation. Okay, change in the DNA. So it's a point mutation disease that I had told you, that results in the missense mutation, which converts glutamic acid, which is an acidic amino acid, to a neutral amino acid known as valine. Okay. Now, the genetic defect adenosine deaminase deficiency may be cured permanently by. Okay. Now you come to the adenosine deaminase deficiency. Question number 50. You can say 5. So activator will not do. It's a gene therapy. It will need gene therapy, right? Adenosine deaminase deficiency will require gene therapy. So what it does is, it is an enzyme adenosine deaminase. First know what is adenosine deaminase deficiency. Okay. So adenosine deaminase, okay, is an enzyme, okay, which involves, you can say, adenosine deaminase deficiency, means deamination means removal of the amino group is happening. Okay. So what happens is, what it does is, it causes deamination. What kind of deamination will it cause? Okay, and which type of deamination will be done by this specific, you can say, ADA. So it is considered as one of the key enzymes in case of purine metabolism, means A and G, adenine, guanine. Purine metabolism. Number one thing is purine metabolism. Okay. So what happens is, you can ADA association, or ADA, what it does is adenosine deamination. It leads to pulmonary fibrosis. Okay, so what happens is it will not be able to deaminate adenosine properly. Okay, so causes pulmonary fibrosis, the fibrous tissue, the collagen development in the lungs. Okay, so chronic exposure to high level of adenosine can also increase or exacerbate or exaggerate the inflammation response. Okay, so that can happen. So we cannot suppress the adenine, right, or the adenosine which is present in the DNA, whatever things we eat. Okay, so ADA, specifically the enzyme ADA1, it is found in lymphocytes, it is found in macrophages, means B and T cells, lymphocytes. Okay, then macrophages. It is found in these areas, or these cells, you can say. And these cells result from where? They come from hematopoiesis, that is the bone marrow cells. Hematopoietic stem cell, I told you, they make lymphoid progenitor cell. From there, these cells will come out. From myeloid progenitor cells, other granulocytes will come out, right? So you need to put it in the bone marrow. Means introducing bone marrow producing cells with ADA. Okay, at embryonic stages, you can go for gene therapy. Okay, then we can go for this kind of, or we can think of the remedy for this kind of specific, you can say, disease. That is a gene therapeutic event, right? So we have to insert the gene either by using a lentiviral vector, AAV adeno-associated viral vector. So those are not in your course, but those are viral-based vectors that we are using. We don't use PBRT22 in case of humans, right? So we use those high-end vectors, but those are not in your course. Okay. So that is done in case of ADA. Okay. So it can deaminase adenosine. Okay, that is the way that this, you can say, function works. Okay, so I think all the questions are clear till now. Now you can practice in your home, or at your home, the other questions, like you have 56 question, quite same pedigree analysis. You can practice these questions. Okay, so similar questions again, you have Down syndrome, erythropoiesis, you can say fetalis coming in the linkage, question number 58, 59. So you can practice these types of questions. Okay, so we'll do some specific questions, okay, which you have not been able to do. We'll be taking some tough questions from this worksheet and try to finish this worksheet in the next NEET class. Okay, like Huntington's disease and sickle cell anemia, question number 65 is a good question. So we'll take those challenging questions from the next class, okay, and finish this worksheet off. So I hope you were able to comprehend the topic. And if you have any doubt, you can ask me in the on my WhatsApp number. Okay, then thanks a lot.